Without further ado, okay, we can start, and that is your slide change.
Okay, perfect. Okay, thank you very much. So yes, I'm here to talk to you about photo ID of harbor porpoises, and as I kind of said before, most of the time I tell people that and they laugh at first. I go, no, for real, I do. So I'm going to go over a little bit about harbor porpoises because they're kind of the underrated guys here in the Salish Sea, and then tell you more about what we're doing and how we're doing it. So just a little recap. I know this is probably, everybody knows this here, but we went to Mount Vernon in Burlington for a festival the other day, a couple weeks ago, and realized that many people there had no idea what the Salish Sea was. So I just thought it was interesting, just going a few miles away, that it was a bit unknown. Let's see, so the Salish Sea, of course, is all the inland waters of Washington and Canada, in a very high biodiverse area, so really a great place to live, to be, and an important one to protect. So what our study site that I'll be talking about is right here, so a little bit north on Anacortes on Fidalgo Island. So, moving on. What is a porpoise? Good question, I'm glad you asked. So that's one of the questions, right? Most people know dolphins, but porpoises, again, under the radar. So Pacific Mammal Research is research and education, so we do scientific research, but we also think it's very important to educate everybody. So hopefully this works. So we just started doing these new little videos. So to tell you the difference between a porpoise and a dolphin, I would like to hopefully do this if it has the sound. So let's see if it works. Oh, just click it. Oh.
One of our most commonly asked questions is, what is the difference between a dolphin and a porpoise? The terms used to be used interchangeably in the past, and while both are cetaceans, which include whales, dolphins, and porpoises, they actually are in different taxonomic families. Let's go over how to tell them apart. Porpoises are in the family Delphinidae. There are 6 different species, and in general, porpoises are smaller, but have a more robust body than dolphins, and prefer colder waters. The example species I will use today is the harbor porpoise. There are a few key features to tell the difference between a porpoise and a dolphin. First, let's look at the most commonly seen part of the animal. Porpoises have a small triangular-shaped dorsal fin. Next, porpoises have a round, blunt mouth with no pronounced beak or rostrum. And if you ever get to look inside their mouth, you will find spade-shaped teeth. They also differ in their behavior. In general, depending on the species of course, porpoises live in smaller groups and are more shy and elusive. Blink, and you may miss them. Dolphins, on the other hand, are in the family Delphinidae. With 33 species, there is a little more variation within this family compared to porpoises. But for most species, the key differences hold true. The example species I will use today is the well-known bottlenose dolphin. In general, dolphin species are larger than porpoises, but with a more streamlined body shape. Now we will revisit those features. The dolphin's dorsal fin is more curved or falcate in appearance. In most species, their head has an elongated beak or rostrum. Here is where some species vary, as some dolphins, for example, the largest dolphin species, the orca, or killer whale, has a more blunt mouth. Inside their mouth, you will find cone-shaped teeth. Dolphins are much more likely to be seen being acrobatic out of water, riding the bows and wakes of boats and breaching, for example. In general, depending on the species of course, they are more gregarious, living in larger groups than porpoises. So now you know the difference between a dolphin and a porpoise. So next time you are out on the water, look for those dorsal fins. Be sure to like the video and subscribe for more, and check out our website for more information.
So we're planning on doing quite a few more of these. We have another seal and sea lion one up on our YouTube channel, but short little 2 to 3 minute infos about the animals in the Salish Sea. So hopefully now you have a more concrete understanding of the difference between a dolphin and a porpoise, and you can tell your friends. It's a nice thing to do at a party, you know. No, that's actually not a porpoise, it's actually a dolphin. So a little bit about the life history of porpoises versus dolphins, because again, most of us know dolphins and kind of the average stuff about them. So let's put it in relation to them. So the harbor porpoise, and again the bottlenose dolphin, because that's one everybody knows, flipper and whatnot. Most of the ones you see in the aquariums. So bottlenose dolphins can get very large, 300 to 1,400 pounds for the big offshore guys. And harbor porpoises are quite smaller, 150 pounds on average, 5.5 feet, kind of average person. 6 to 12 feet for the bottlenose dolphin. So you can already see, much smaller. The lifespan is an interesting one. So most dolphin species live 40 to 50 years. Of course, the orcas are a little bit different. But we had really no idea exactly how long porpoises live. If you look at the literature, there's range anywhere from 8 years old is the max to 25. So kind of a large range, but the key thing to keep in mind here is that it's much shorter. It's about half, at least half or less than half the time of dolphins. So because of that, if you have to be alive and make babies, you better start doing it earlier if you're not going to live as long. So sexual maturity is pretty quick for harbor porpoises at 3 to 4 years of age. And again, you've got to get those babies out. So give birth every 1 to 2 years instead of every 3 to 4. So that's the basics between harbor porpoises and dolphins in general. So next we'll talk about the two species that we see in the Salish Sea. So the first one is the Doll's porpoise, and we don't see as many of these now as we did in the past. You saw a lot of dolls and not a lot of harbor porpoises, and then they kind of switched. We don't know exactly why that happened, but it is what it is. So here is the sound, hopefully. It's quiet. Sounds like someone tinking a glass. So now porpoises speak in the ultrasonic range, so we actually can't hear them. So what they've done is for this one, they've slowed down the recording so we can actually hear it. So the Doll's porpoises are black and white. They're much more splashy when they surface, so it's a characteristic what they call a rooster tail splash, because I guess somehow that looks like a rooster tail. But very splashy when they come to the surface, and black and white, so sometimes they get mistaken for baby orcas, but of course they aren't. Next is the harbor porpoise. So these guys, very much more sleek in nature when they come to the surface. Again, blinking, you may miss them. So here's a typical surfacing. So we have one, there, there, and there. All right, so pretty, sometimes it doesn't even look like the dorsal fin comes out of the water. And so here's a sound of them foraging. So you hear the clicks. So it slows, and then when it goes, that's when they've kind of targeted in on a fish. Again, both of and this is the clicks, both of these have been altered so that we can actually hear them in our range. So this is more of the, similar to the Doll's porpoise. So it's pretty cool we have the technology now to be able to record them in the first place, and then be able to adjust it so that we can actually hear it. So a little bit about harbor porpoises in the Salish Sea, and hopefully this will set up well for the next talk, talking about diseases and stuff in harbor porpoises that Stephanie's going to do. So in the 1950s and 60s, they were really abundant. They were everywhere, right? Lots of harbor porpoises. But for whatever reason, they were all but gone by the 1990s. And again, we don't know why this happened, and we don't know why recently they came back. So in 2016, they did some aerial surveys, and they estimated that in the Salish Sea, not including all of Canadian waters, was over 11,000 individuals. So this is one of those unique, like, great stories. Right? There's a lot of sad stuff going on right now, but this is a good one. Right? They're doing really well. And they think that perhaps there was pollution and the gill nets and anthropogenic interactions, people along the coastlines doing work and things, has probably contributed to them not being here. And then the fact that we're trying to make that better is maybe allowing these animals to come back. So it's a great story, but we don't know much about them. So that's really, we need to change that. So you might be asking, like, well, I don't care, what? I mean, we all care, but many people are like, well, why do we care about harbor porpoises, whatever? So they can be very good sentinel or indicator species as being top predators, and that they live in the waters year-round, and they are sensitive to environmental and anthropogenic changes. And kind of because they do, their lives go much quicker than some of the other species, you can see changes in their population a little bit faster. So they would be really good if we can monitor them, and if their populations are suddenly going down, then we know that there's something going down wrong below them in the food web. And so we can start to try to look at that earlier. The other hand, we just heard from Howard that transients are a whole bunch of them are around now, and they're increasing. And what that means is that they eat mammals. So they're eating harbor porpoises and harbor seals. So they're prey for those guys. So if we want to make sure that the transients are doing well, they also need to have food, just like the southern residents do. So as much as I don't like thinking about porpoises being eaten by transients, it's part of life, and so we want everybody to thrive. And so we need to understand more about their numbers and where they go and why they're here to be able to help them as well as the other species. So here I'll step on my soapbox for just a minute. Long-term monitoring is crucial, right? We know the stuff about the orcas because we've been watching them for decades. The Wild Dolphin Project in the Bahamas, spotted dolphins and bottlenose dolphins. We know everything about their families because we've been monitoring them for 30 years. But the unfortunate thing is long-term monitoring is not really sexy to funding. So it's really difficult to get people to no, we need 10 years of data to be able to tell you the answers that you're asking for right now. So it's just really important for everybody to support those that are doing these long-term monitoring studies to get us the information that we need to be able to make the actions that we need to make to help them. So even though all these things are true, we know not very much. So we know that there is a stock, the Washington inland water stock, which is one that I'm be talking about, the Salish Sea one. And then there's a stock along the Washington and Oregon coast, and they do believe that those are genetically separate, that the inland waters stay here and the ones in the offshore stay out there. So we know we have relatively unknown population structure, abundance, distribution, movement patterns, ecology, and behavior. That's kind of everything. So it's really a huge lack of knowledge that we have about these guys. And they are most likely, if you go out, they're the animals that you're going to see, if you see anything, because they're one of the most abundant marine mammals. Basically, probably harbor porpoises and harbor seals are everywhere. So, for whale watching books and things like that, let's support these guys. These ones you'll see, and they're cool. They're cool too, and I hope to convince you of that in this talk. So why do we know so little? Well, that's a very good question. And again, I'll go back to the blink and you may miss them. So if you've been out in the water, you've probably seen this. Oh, there was something. Let's watch it come up again. No. Just kidding. Yeah, so you can see the animal surfacing just under here, and this is where he's just gone. So it's a great way to tell us where they've been, not necessarily where they're going to come up, unfortunately. But they're just harder to study, right? Because they are relatively more elusive and not as gregarious and friendly, we like to use that term, as other species, they kind of get well, there's other ones that we can do. So, on that topic, other marine mammals are slightly easier to research. So, are you more likely to notice that guy? Or that guy? Right, 6 foot tall dorsal fin or 3 inches? Not enough. So and it is important obviously for us to understand the orcas, and they are amazing creatures. But unfortunately, the harbor porpoises just kind of get thrown to that wayside because they are harder to study and they're just not as exciting as some of the other ones. So what do we know? We do know a little bit. I know I said we know nothing, but we know some. So we do know that their movements are linked to productivity. So these guys have a really high metabolism, so that one study was showing that if the animal is already not doing well, they can actually starve in as little as 3 days. So most of them can go a little bit longer than that, but that's pretty short compared to other marine mammals that can, you can not eat for a while. These guys can't. They really need that predictable food sources. So that's a big deal, right? Especially with what we're, all the changes that are happening with forage fish and everybody being able to find food. These guys really have to be able to know where it is and have it be there when they get there. So lots of times we'll see the harbor porpoise here, and of course these are the seagulls. Seagulls are kind of mean. They're surviving. But they take advantage of the harbor porpoise in that they've just, try to catch that fish between the surface and themselves, and then the seagulls like to take it, unfortunately, but not always. So, and all the pictures that you'll see here of the porpoises are all pictures that I've taken, or my assistant has taken in Burroughs Pass, where we do our research. So the other thing is sociality. So we know that they do vocalize with clicks. Other dolphin species use whistles. So you may have heard signature whistles as a name for individual dolphins. These guys don't seem to have that. So, and because they live in smaller groups, right? These guys are in groups of 1 to 3 animals, and dolphins are generally 5 to 10 or more. They basically were like, nah, they're just not social. They're asocial, right? Asocial means they're not social at all. But it's been more debated, right? They have some in captivity that have shown like, no, they do play. They do play with each other. There's large aggregations that we're still trying to figure out why they occur, of 50 to over 100 animals. You know, why are they getting together? And there's a new paper that just came out, really exciting last year, that talked about the click trains that they have, and actually documenting that they basically are using those as social interaction. So they're using their clicks not just to find food, but to interact with one another. So that they are more social than we believe them to be, or what we can see from the surface. So that's kind of exciting. I've always thought that there's more to them, but it's nice to get some support for that. And reproduction. So generally in the summer, late summer is when you start to see babies. In our area, August is usually the earliest time we start to see cats coming in, and the most likely time we'll see cats is in the fall. We did see one as early as May one year, which is a little odd. But this is, of course, a mother and calf here, or we assume is a mother and calf. And this is some little reproductive time happening. We'll get to that later. There's more exciting photos. Just wait. So we don't know, we know the basics, right? That's when the calves show up, but we don't know where they go to mate or when they're exactly doing it, where they do it. It varies by location. There's a lot of research on harbor porpoises in European waters, in the North Sea and the Baltic Sea, but less so here. There are some areas in the Salish Sea that they've documented. The Anna Hall up in Vancouver looks like that might be an area that's important for reproduction, but we're not positive. So how do we study them? Well, they've been basically studied by aerial surveys, and these are great. They give you abundance estimates, but it's a snapshot in time, and they do fancy math to, they count the individuals and then do fancy math to tell you how many you missed. So it gives you a nice snapshot in time, but nothing about throughout the year or even by day. So limited information. Strandings, we get a lot of information from strandings, which are great, life history and physiological stuff. But again, that doesn't tell us about their everyday lives and what they're using and what they're doing. Side note, if you ever wanted to sex a dolphin or a porpoise or a whale in general, I believe this one's a female. A female will have a long slit here, a long urogenital slit, and then a slit on each side as their mammary slits. But a male will have one long slit and then a tiny little anal slit in the bottom. So if you think about it, females have a division sign and males have an exclamation point. Which totally makes sense, right? So that's another good one for parties that you guys know how to sex a dolphin? I do. Unfortunately, you have to see their bellies to do it, so there's that. Females are a little bit bigger for harbor porpoises, but not in a way that you would be able to go, oh, that's a female there. It's just a little bit bigger. Passive acoustics, or acoustics in general, have been used quite a bit, but that's only been recently for harbor porpoises because, again, they vocalize in the ultrasonic range. So for a while, we couldn't even record the sounds. So in the last 10, 15 or more years, they've been able to develop things that you can actually record, the ultrasonic vocalizations. And the other issue with recording is that, this whale is saying, he's like, hey, dude, we're over here. Right? You have to put it where they're going to be vocalizing. And the issue with harbor porpoises, too, is because their frequency, the high frequency that they have is narrow band, so it's very directional. So you have to be kind of right in front of the microphone in order to hear it. So it's great information. You can get it year-round. You can put passive acoustic monitors out and just set it and forget it. let it out there and you have data for months, but it has to be in a good, right place and they have to talk into it. This is our boat. And I don't have a picture of us on the boat yet because we're still getting it fixed up, so I put my assistant from another boat on top of the picture of the boat in my driveway. So that's what it'll look like when we go out. But traditional boat-based surveys are what most people that do cetacean work do. You go out in a boat, you take pictures, and you go zoom around. A little bit harder with the porpoises because they don't love boats as much as others. And I'll tell you that that's not totally true. We're finding, but they aren't going to come and ride your bow and be like, hey, look at me. So that's been it's a little bit harder to get information from them sometimes that way. So the other idea is to do it from a land-based study. And so this is my assistant Katrina and I here, out on our little rocky outcropping that we spend hours at a time out there, and usually in the cold. And so this is a great one because we don't have to worry about them being afraid of boats. But you do have to have a place where the animals come close enough to land. So luckily we did find a place that happens with harbor porpoises. So that's where we started our study. Like, well, let's see if we can do it from here and then expand it from there. So Burroughs Pass, if anybody's been up to Washington Park in Anacortes, it's a beautiful park. If you have never been there, please go visit. You can basically year-round see porpoises and or seals there. We did have somebody, one of our citizen scientists said they saw a humpback whale go through there once. Pretty sure that whale was lost. Happens. We did get to see some transients come in last year. It was very exciting. My assistants and I got really excited. But they came in once, so you'll occasionally see other things. But so this is from the top observation outlook, so a little triptych picture of the pass. And there was another project that was being done previously before we got here, the Harbor Porpoise project by the Pacific Biodiversity Institute. And they had documented that, yes, this was a high abundance for harbor porpoises. They were there year-round, and you could almost always see them at some point. So they did a lot of that base work, so we said this is a great place to start to try to get pictures. So here we go. Here's the porpoise swimming into Burroughs Pass from Rosario Strait over here. And so this is Fidalgo, so you come in here and go all the way out here. This is Washington Park here, that Skyline Arena, if anybody's familiar with that. So that's where we hang out and sit. And so why should we try to know these individuals? Well, I mean, what we know about the orcas is because we know the individuals. What we know about the dolphins in the Bahamas and in Australia, in all of those places, is because we know the individuals. So this is from a pinniped branding thing from NOAA, but it holds true no matter what way you're identifying them. So by reciting individual animals throughout their lives, we can learn about age-specific survival, migratory movements, habitat use patterns, human interactions, resource conflicts, site fidelity, age, sexual maturity, reproductive success, and longevity. So basically all the stuff we didn't know, we can find out. And especially with social animals, which I do believe the harbor porpoises are, maybe not quite in the same way that dolphins are, but understanding their individual lives and who is interacting with who and why they are really tells us a lot about where they're going to be going feeding, who's going to be doing that. Is there disease transmission between different groups? Are they smaller communities that are separated, or is it one big population that has just a big mix? We don't know, and that's really important for conservation. So knowing the individuals really helps us understand all of this. So one of the coolest ways to do that is through photo ID of natural markings, and it is non-invasive. So I spent over 10 years working with Denise in the Bahamas, researching spotted dolphins and bottlenose dolphins here. This is Cooper. You can see he's kind of beat up, the male. This is Brush over here, I believe. And so I got to know these individuals and their kids, their calves. Orcas, of course, they've used humpback whale, the coloration patterns on their flukes and the trailing edge shape, the spotting patterns on cheetahs. You can do it on elephant ears. It's kind of cool. And the most surprising one, when we were working up our study, my assistant found a thing that you can do it on painted crayfish. Who knew? So anything that has a stable mark, some kind of marking that is stable over time, you can use that. So you take a picture, and then you take a picture another time, and now you have this long-term set of, I saw them here, I saw them there, they were doing this individual was doing that, this one had a cat, this one didn't, etc. So, again, do it on harbor porpoises. Like I said, I usually get laughed at, because yeah, sure. Like usually, this is the kind of picture maybe you'll get if you don't just get that picture. And there is a harbor porpoise here hiding under the wave. So it's really been attempted for all those reasons, right? They don't seem to have overt markings, like they're just kind of darker, and they don't have a 6-foot tall dorsal fin, so it's more difficult. Their dorsal fins don't seem to get messed up very much. So it's been really been attempted in the past. And so we were like, I'm gonna try it. Let's see if it works. So the one thing that really helps with this is a really long lens. So this is a 100 to 400 millimeter zoom lens that I use on my Canon T3i camera. And so this really helps. Now, even when all those pictures that I'm showing you are taken with that lens and then zoomed in on the computer. So remember, I'm like hundreds of yards away and the dorsal fin's like this big. So, but the technology now is good enough that we can get these pictures, like the digital age, right? Having such high pixelation that we can get down to allows us to take pictures from farther away but still have the resolution available to do it. Back when I had to do slide film, that probably wouldn't have worked. And I worked with slide film. I spent hours with my eye on this little magnifying globe looking at 35 millimeter slides. And it was much nicer when I switched to digital. But so we were able to, so we went out and we're like, let's try it. Let's see if they have markings. Let's see if we can do this. And so long story short, you can. So to this date, we've been doing it since 2014. We have over 70 individuals that we've recited in our study site. So it's always exciting when you get a reciting of an animal, but it's a little bit more so in our office. Yes. So we have a whole bunch, and I'll just note Jaws up here is my assistant's favorite and kind of mine too. Obviously we named them to help us with their markings, but we just thought the coloration on that just kind of looks like a great white shark. And what's even cooler is that we're pretty sure that this is a female. So, yeah, don't mess with her. So we, as Howard was saying, it takes a lot more work to ID these guys, but it's so rewarding, and we're learning so much about them. So about 35% of the animals that we have been recited in the past over months, weeks, months, and years. So these animals are coming back to this particular area, which is really important. It's very, very exciting. So I'll tell you a little bit about how we do that. There's a lot of stuff. So we have a 8-category matrix because, again, most small cetacean studies use just their dorsal fins, but that's really not an option for us. The minke whales actually use the same thing that we're kind of doing. So I saw that and I was like, oh, that's a great idea. So the minke whales use the coloration patterns on the side of the animal, and that's our main thing that we use. So the pigmentation patterns along here, so this is Jaws again, you'll see him with her a couple times. So there's different variations. So this is kind of marbling and blotchy coloration. This is a specific two-tone coloration. And we're noticing that those markings are individual by individual, and they are stable over time. We use scars and lesions, so unfortunately for them, but fortunately for us, they get scars and they survive, and they have them left. So there's one here, that's Bolt, this is Dodge here. So those are helpful for us. On their peduncle, so the area between the dorsal fin, the back of the dorsal fin and the fluke, we use. The trailing edge of the fin, so there are a few that do have nicks and notches in their fins, and we love them because it makes it a lot easier. So this is Nip. She was one of the first ones. We believe this is a female as well. She was one of the first ones that we identified, and she's been seen every single year since the beginning. And then we do confirmation marks. So the primary and secondary marks are the ones that we would use to actually ID the animal, and then we use these other ones to help confirm, just to make sure that we're seeing the right animal. So we have fin shape, fin size, fin base width, and overall coloration. And I'm going to go through each of these just real quickly. So the pigmentation, we have a uniform where it just basically looks all gray above and below that kind of lateral line of changing coloration. You have Jaws here again with our distinct two-tone coloration there. The marbling and blotches, so this is Lightkeeper. She's another one I don't know that, where the line isn't quite as drastic. It kind of marbles into the other. And then some that have just very distinct white patches. This isn't the greatest picture, but you can see there's a white, like giant white patch that's there. There's not really that many of those. Scars and lesions, so this is an interesting one. There's quite a few. So this is tadpole down here. This is Flash Gordon. I'm kind of a sci-fi comic person. I think that's Bolt again. This is Dodge, and the reason why I named this animal Dodge is because it looked like it dodged a bullet. She's got marks over here. There's puncture wounds here that we don't know what those are. We have scars over here. Like it's made like all over the place. So not sure what was going on with that one. But also documenting these things, we can try to figure out what those scars are from. Why are they getting them? Is it from anthropogenic stuff, people, or is it just from whatever they're doing under the water? The trailing edge, of course, we have some with a notch in the upper, we have some with a notch in the lower, and then some with a notch in the middle. And this was given to us by a whale watching boat that was able to capture a picture for us. We don't have any that have nicks and notches all the way up and down. It's crazy. The peduncle, so just that peduncle area, so we have scars on the side. We have, this is nip again, and we'll have a better picture of this later, but there's scars all like indentions all the way down the peduncle. Yeah. Overall coloration, so we do kind of see, and this does depend on the lighting that you get with the pictures. Some that are really more a silvery gray, some that are more brown, some that are kind of that in-between gray and brown, and then of course that two-tone. So you see this one is crazy. I think that one's snow because it just looks so white. So the fin shape, and this was one of the interesting things that we've discovered, is that there seems to be a certain number of shapes, general shapes that we see in the dorsal fins. So within that, mainly triangular, not falcate like the dolphin. So we have a leading bump, so there's a big bump in the top edge, the front edge of the fin. One that a hook tip, so it kind of just looks like it curls just over at the top. Pointy, just like a pointed hat. The more basic triangular shape. And one that's looks more like a dolphin dorsal fin, so more curved or falcate in nature. And we can see that most of them, we do have another category if they don't fit into those, but generally most of them fit into one of those categories, which is kind of interesting. So fin size, we also looked like some look really tall. Again, we're talking 3 or 4 inches here, so it's all relative. So super tall, kind of average what you would think, and then little stubby guys. So that's Lightkeeper again, and it just looks super tiny and cute. So now the base of where the dorsal fin meets the back is really interesting. So some are really wide. So if you were to take where the dorsal fin ends and the back begins, to be like there and there. So very kind of wide base. And then you have one that's kind of average, right? So a normal looking dorsal fin. To me, this one just looks like spread out like that. We don't really have any that we would consider narrow at this point. I don't know if those exist. So those are the marks that we're using. So what are the challenges? Well, there's a lot. But the first thing is, of course, proximity. So to be able to be close enough to the animal. So that's a porpoise right there. So from the same pictures that I was just showing you, I also get that picture. So it's important for them to be a little bit close along with my zoom lens. Amount of body above the water. So because we aren't just using the dorsal fin like other research groups do for other cetaceans, we need to see that side of the animal. And like I said, sometimes they are not very helpful with that. They just don't come out. But more often than you think, they actually come far enough out of the water that we can get most of the side flank of the animal. The biggest issue is the erratic surfacings and lack of repeated ones. So as I said, they'll come up, and you go, okay, great. And I need you to come up again so I can take your picture. But they come up, and then I'm like, all right, I get it, I've got it. And then they come up over there. So it's a little frustrating. But if you're out there long enough, a lot of times they will end up going, okay, I'm gonna come up 2 or 3 times in a row, and then, my assistant and I are like, thank you so much. So it's about sitting out there and waiting. But that is the biggest problem, is they just don't tell you where they're going to come up next. And of course lighting, because we are looking at the coloration of the animals, beautiful backlit sunshine wonderfulness is not going to work for that. We could get the outline of the dorsal fin, but that's not as helpful for us for for that. So luckily, some of that can be changed in post-production, as we like to call it. So take it back to the computer and adjust the lighting there. We can oftentimes get some usable photos from otherwise looking non-usable ones. So we take all these photos, but we don't just take that information. We also take all the environmental information, group behavior, where they are in the past, are they using one area more than others, calves are present, behaviors, their state, are they foraging, are they traveling, etc. And any notes or interesting things that happen, because sometimes things do. And when we do, they get a lot of explanation points. That's our assignment. So putting it together, we sit out there. Most of the time we look like this picture, but occasionally we look like that picture. And we roll up our pant legs, like there's sun, it's awesome. So we live for those days, but most of the time we're out there in our caps and our jackets. But it's still a beautiful place to be even when it's cold and you can't write. So we take the pictures. Side note, we also do this for harbor seals. So we take pictures and do photo ID on them. It's a little bit easier because we just have to look at spotting patterns for them. We're working on a software program that has been slightly difficult in working with us. But we're going to hopefully be getting that up this year and be able to run through our photos and see who comes back to the pass. We do already have some identified individuals that come back to that area, so it'll be exciting. Then we take that information with the environmental stuff, and then we make publications. So our first publication on our last 4 years of work came out in 2017, the end of 2017. It's in Marine Mammal Science. And if anybody's interested in it, you can not get it on our website yet. I think you still have to email me because it's still on embargo from being in the paper, in the journal. So you can email me and I'm happy to send that to you. But if you just want the recap, I'll tell you that now. So that should be good. So this is for the conference, a Marine Mammal Conference in 2017. Looking for confirmation from my assistant. We got a presentation, and they were doing this new thing, doing video presentations, because the scientific community is trying to get better at relating information to everybody and not just the scientists. And so they were trying this new format, doing a 4-minute video. And so we were excited because they got the confirmation, like, awesome, we got a video. Oh my God, now we have to make a video. And, okay, so, and we had to do it within a couple months' time, and then they had to send it in, and then they said if it was okay, and then you got to do it, and if it wasn't okay, then you did a different presentation. So it was a little bit stressful, but we had a lot of fun doing it. So we had to cram our entire paper, and the 4 years of work that we did into 4 minutes that was accessible to scientists as well as everybody else. So this is the end product of that. It's about, again, 4 minutes. So this will tell you all the data that we've collected over the last 4 years.
The harbor porpoise is one of many cetaceans we have in the Salish Sea, but one that is often overlooked. They were all but gone by the 1990s, but have happily recovered and are once again one of the most abundant species in the region. We don't know exactly why the decline and recovery occurred, but we also know little about their long-term population dynamics. This is crucial to understand for future conservation efforts, and we here at PacMam want to know what are their group dynamics, behaviors, ranging patterns, and social structure. These are all questions we hope to answer, knowing that the basis for improving our understanding of a species is founded in the identification of individual animals and tracking them over time. Burroughs Pass off Fidalgo Island provides the ideal location for this type of study, a land-based point in a semi-protected area with year-round presence of harbor porpoises. It is a tidal stream habitat where large tidal flows pass through a narrow strait that often provides predictable and favorable foraging conditions. This habitat is likely very important to this small species, which is limited in the amount of energy they can store, making predictable food sources critical for their survival. Photographs and environmental information were collected from an observation point about 6 meters above the water during 1 to 3 hour observation periods. Photographs were compared using a matrix of 8 different identification marks, and presence and group size were analyzed in relation to variables including season, riptide strength, which we call RIP, and behavioral state. Through photo ID, we have begun to know the individuals that use this area, gaining insight into their daily lives. Calving appears to occur during late summer, with calf sightings peaking in fall. We were excited to discover that many animals continually return to this area. Limited information suggests that harbor porpoises have relatively small home ranges. Thus, our work indicates that they may have a more fine-scale population structure than previously understood, and that it is influenced by season, behavior, rip, and calf presence. However, However, unfortunately for tourists and researchers in Washington State who enjoy sunshine and dry weather while observing porpoises, there are fewer animals present and they are in smaller groups during the summer months. We found that they usually travel in small groups of less than 3 porpoises, and that group size increased with calf presence. Groups were also significantly larger when foraging compared to other behaviors. In this habitat, there may be an advantage to having greater numbers when feeding, even if they are not actively working together. Harbor porpoises, like us, will go where the food is, so these seasonal shifts may indicate changes in prey availability during the summer. But following prey into tidal stream habitats can be tricky, as the water can be turbulent during times of high rip. We found that porpoises were more likely to be seen in low or medium rip, supporting the idea that they prefer low energy areas, but may gather in high energy areas when foraging benefits outweigh the energetic costs. These costs are higher for immature porpoises, so mothers and calves will avoid strong tidal flows. Indeed, we found that calves are more likely to be seen during slack high tide, when water movement is greatly reduced. Our study has shown that season, behavior, rip, and calf presence influence group characteristics of harbor porpoises in Burroughs Pass, and supports the idea that there is a complex relationship between habitat use and population structure that varies on small spatiotemporal scales. We show that photo ID is possible on this species. There is likely a local population of harbor porpoises that utilize Burroughs Pass, and this may be a biologically important area for them. Even for populations that are stable or increasing, understanding fine-scale population structure and collecting baseline data is crucial, as all species are still subject to changing climates and anthropogenic interactions. More detailed research such as this study will contribute critical information on harbor porpoises and will be vital to fully understanding and ensuring the protection of this species and their habitats.
And I'd like to note the underwater video footage is not mine or ours there. Florian Granier was nice enough to give us his footage for that. So we're very thankful for that. And thankful to my assistant Katrina who did all the editing for that video. So we found out some really cool stuff, again, by knowing these individuals. And so we're looking forward to doing more. As you can see, from that one, we had 50 animals at that point. We're up to over 70 now, a little over a year later. So we're excited about the future. And we have found out some really interesting information. So some interesting insights. We do have those highly scarred individuals. So again, you can't see all of them here, but it's a nick, big gash, nick, giant gash, right before. So we don't know what happened there, but kind of looks like a propeller, right? But she survived and is doing pretty well. We saw her last year. And then of course, we have Dodge here again, those weird, strange marks that we're not sure exactly what they are. So there's where these scars are coming from. That's something we want to hopefully be able to find out. In contrary to popular belief, they do wake ride. Now, I've never seen them bow ride. That's a whole different bag of fish or whatever. They do wake ride though, so it's pretty cool. So he's there. So it's pretty cool to see. Now, what they do is usually, so the boats are coming through the pass. So they're not like following you on the wake, like a lot of dolphins would do. The boat goes through and is probably most of the way through the pass, and then they're like, okay, it's safe. And they ride the wake after the boat has kind of gone through. And we see this quite a bit of time. We have quite a few pictures. This is obviously the best one. But it's really fun to watch, and you'll see some of the younger cats do it too. They're like, whee, like down the side. Okay. Now, this is where I want to put a disclaimer. This is where we're going to get a little bit more graphic. If you've never seen a harbor porpoise reproduction event, it'll be a bit drastic. So we do see reproduction, and we do see it year-round. So this is the interesting part. All of these pictures that you'll see, we have taken in every season. So they're doing these mating attempts all the time, so some of it's probably practice. And so this first one, we took this in our first year, and we were like, oh cool, we saw them come out of the water. Yes! not realizing what we had actually seen. And we went to a conference in 2015 in San Francisco and worked with the Golden Gate Research down there, who's the only other group besides ourselves that we know of that is conducting photo ID on the species. They do it from the Golden Gate Bridge, which is pretty awesome. We got to go on the Golden Gate Bridge and they just come under with the tidal rip and they're just down there eating. So they have a lot of great video and stills of this, and particularly reproductive events. So what we found out there was that what they were seeing is that it was always a male coming up on the left side of the female and oftentimes jumping out of the water, but it's always on the left side. They're very handed in that respect. So you can see here that this is the male coming up to the left and the female, that's her fluke on the side. So she's like, and they gotta do that as they go. So I went back when after we learned this, I was like, wait a minute, let me look at this photo again. I think we actually caught it on camera. So we were excited about this. This was great. And then this one, then we got this one. We're like, oh, yeah, that's okay. That's definitely. Now you can see the female on this side over here, and the male's coming up this way. But we're still at proof of it. And let me just put a disclaimer here too. I came from spotted and bottlenose dolphins who are not as well endowed as harbor porpoises. So even if you caught it in the air, you might not see it, especially spotted dolphins. But see if you can start to see it. Now this one might be hard because you can't see the color, but there's something right here. And again, right side, this is the female. The male is coming up and out from the left side of the animal. Now, you can probably see it there. Again, female to the right, male coming up. That's what that is. Wait for it. Wait for it. Hello. There we go. There's another one that's better though. Just wait. So this one was really far away, so it's not a great picture. But I realized, again, I went back and I was like, wait a minute, I can see it. Right, so we get very excited. So there's this one. So then we were taking pictures and I got super excited in the field because I'm like, I caught them in the air. So to catch them in the air is just hard to do in the first place. So I was just super excited. And thank goodness for digital cameras. I was like, Ken, I think I got it. I think I got him in the air. And so I'm zooming in on the camera and I go, oh my God. I got that. So, it's all the way up to their chin. So it's pretty impressive. And the first time that I saw this, I was like, that can't be real. This is Photoshop. And then I took a picture, I was like, nope, it's true. So I was very excited to get this one. So we're definitely, so all these other ones that we're seeing, and because of that directionality, we're 99% sure that those are mating attempts. So this was a very exciting day in the field, needless to say. And we learned more from the conference 2 years ago. Another researcher from the Golden Gate Research in San Francisco was doing stuff on the reproductive structures in the males and females. And so they actually did a mold of the uterus and the vagina and everything of the female, and then took inflatable guys, because you can't get a real one. So they use these inflatable ones, and we're able to show that they evolved together, and so that directionality is on purpose. Like that's the best way for that to go into the female, is that left-handed side. So that was really interesting. So we're learning lots of cool stuff about these really cool guys. And it is very quick. It's just, and that's it. And then they probably don't do it again, or at least in our area. So it's really exciting to get these kind of pictures. So that's my most exciting picture. So what does that mating lead to? Well, it leads to babies. Cute babies, yes. So little calves coming here, and you oftentimes see them just short into the back of the female. That's called echelon position. So they kind of catch the ride of the females, kind of a little wake. So it's a little bit less stress for the calf to go through the water, gives them a little break. But one unique thing I want to mention is that they'll do what we call chin slap breathing, where when they come, you see all the surfacing, they come up real nice and just like that. Babies don't know how to do that yet. They're kind of awkward. It's like, babies learning to walk. They're not like, I've got this walk thing down. They're like, oh, it's all terrible. So the babies will actually come up and take their entire head out of the water to make sure their blowhole gets out, and then slap their chin down to breathe. So that's what this guy is doing here. So this is the head of the baby coming out of the water. So if you see that, a kind of big splash as it's going down, that's most like a younger calf that still hasn't learned that smooth roll quite yet. And here, I love this one because you can see his eye right there. Super cute. So they also need to eat, of course. So we do see a lot of foraging in our area. Again, it's a high tidal flow, as we said in the video. So there's a lot of highly productive area. And again, it's really important for them to find those reproductive, highly productive areas where there are fish that they can get reliably. So this is what we call a surface chase. So they'll come up to the water and then kind of skim the surface and the water goes by. So again, they don't always just not have splashes. They do. And a lot of times you'll see this behavior. So the next one is cool. So this is one coming up just before it goes to a surface chase. And so he's come up out of the water, and I like that one because it looks just like a blanket of water. You can see that it hasn't broken the surface. And then you can see the fish. I made it! I got away! I made it! Oh, no, I didn't. And I have many pictures of this where the seagulls come down and end up coming up with the fish. So I'm sure the porpoises are a little bit upset about that, but survival of the fittest, I guess. So oftentimes they do get the fish, but sometimes they don't, of course. So the next step is what we want to be is the place that everybody sends their photos, so that we can have a kind of a centralized database of these individuals and these animals. So we have a lot of colleagues around the area that are sending us photos. So Marine Education and Research Society, Porpoise Conservation Society in Canada, and the Porpoise Conservation Society with Anna Hall is one that we are working directly with to institute a photo ID program up there. So they have a lot of photos that they've taken. They're going through and looking at all their photos and cropping them for me, and then going to send them in, and so we can start to get some IDs done there. And whether we see a match between them or not, either way is really important information. The Whale Museum, Stranding Networks, Eco Marine Institute down in Puget Sound, and Port Townsend Marine Science Center as well sends us photos from volunteers and whatnot. So we have a lot of groups, and the more the merrier. The more we want to collaborate with everybody that can send us photos and sightings, because the more we learn about these guys, the better off we're going to be. And we do have naturalists, public, we have a couple photographers that go out with not giant lenses. So occasionally you can get them with some shorter lenses if they're close to the boat or the area. So we've matched individuals from their pictures to ours. So we've gotten new sightings of individuals from their pictures. So you can send us sightings, and we do have a booth outside, so you can take a leaflet which has all this information on it, my email and everything. We have a sightings paper that you can take out and then just send it back to us, either in snail mail or email. So you can do it that way. There's also a program called Whale Map, MAPP, and that's one where you can upload any sighting you have of any marine mammal, and it goes into this database. And the great thing about that is the database is open to everybody, scientists and people alike, so we can just pull the data off and use it. So feel free to send us sightings anytime, even if they're not photos, right? The more sightings we get of where these animals are, because people ask me all the time, well, where do they go? I don't know. So we're trying to find out. So we're, like I said, very excited about having our boat. We got that donated last year, so we're super excited and thankful for that. And so we'll actually next time have a picture of us on the boat in the water. Right now it's in my garage. But we do find with the boat-based work and we're working on with Cascadia Research Collective as well, looking at their behavior, and because the animals that we see that are island associated and habituated to people more so than the offshores aren't that scared of boats. Right? If you idle and you stop, they've had them come underneath the boat and pop up on the other side. I've had plenty of times when we've gone out with volunteer people who have been great enough to let us go on their boats with them, that they come fairly close. They're not that scared. So it's another avenue that we have to work with. And this will tell us more about, okay, outside of Burroughs Pass, where did they go, what did they do? We can get more pictures and whatnot. And then we also want to put a passive acoustic monitor out. So to learn about the sounds that they're inundated with every day, as Dana talked about earlier, with the orcas, is loud. And there's boats coming through this pass all the time. In the summer, we have up to 60 or 70 in a 2-hour period. So what is that base level that they're getting at? What are the other animal sounds that they have? And also to be able to record their sounds and understand more about what they're doing in the pass besides just what we see. And the great thing about our area is that we can correlate what we see observationally with the sounds that we get from the passive acoustic monitoring device. So we can go, oh, we were out for this 2-hour period. These are the sounds that we heard. This is what we observed them doing. And we can make much more sense of what is happening. So it's unique to be able to do that. Usually you get one or the other. So on, like I said before, our education, we do a lot of festivals. We give presentations like this. I go around to everywhere from pre-K all the way up to 100+ old individuals in retirement communities. Everybody. And so we do a lot of different workshops and presentations, and it's all free. So if you have anybody that's interested in having us come speak, please email me and we will be able to set that up. So we collect our critical scientific data, we provide educational opportunities to improve the stewardship and conservation of this species and the whole Salish Sea ecosystem. So we need your help. You can volunteer. We have volunteers that can come out and you can just hang out with us for the day out in the field. Sightings, photos. So again, if you have just sightings, it's great. If you have photos, even better. But both of them are really, really important for us in creating that big database that we can have, like the Orca Network has of all the sightings in the Salish Sea of the orcas. We can have that for harbor purposes. And also funding. So if you feel like donating anything, we're also happy to take that as well. Or you know of anybody who really likes porpoises or dolphins, and just don't tell them the difference. So at the end, what we found out is photo ID of harbor porpoises and seals can be done. So that was kind of the big thing. Our paper was really the first one out there that says that you can do long-term photo ID and document it. We do believe that it's a biologically important habitat, that Burroughs Pass. They bring their calves there, they feed there, they mate there. They do all, those are basically the 3 things that are for important parts for a population. They do all of them there. So maybe this is something that we need to protect a little bit more than other areas. And that if we can understand more about what it is about that area that they go to, we can find other areas in the Salish Sea that are like that we'd be likely to find animals, and then maybe set up research there to be able to learn more about what they're doing. Citizen scientists and naturalists can contribute important data, so please help us out with that. And of course, education is crucial for conservation. And lastly and not least, long-term monitoring is critical and funding is needed. One thing I want to know about this picture, which is cool, is we were like, what are those cool markings of this animal? It's really neat. And we looked, like, those are the clouds. Reflecting on the water that's on the side of the animal. Like, so cool. So we found lots of cool things with these guys. So that's the end. I hope that I've convinced you that harbor porpoises are super awesome and super cool and need to be researched more. And you had some fun. I did. So I love sharing the information that we gather. So I'm happy to take any questions, and thank you very much.
So we frequently see a small group of harbor porpoises in Saratoga Passage off Crescent Harbor. Can we assume that those are the same porpoises?
So he's asking, they see a bunch of porpoises in Saratoga Passage, can you assume that those are the same ones? Unfortunately, we have no way of knowing. I mean, when they come into our pass and we don't see them for a little while, and then somebody else comes in, we think it may be the same animal, but we can't even be sure then. You would really have to have the pictures. But we do see, what I believe from what we've done, and this is completely from no actual hard written data, but that these animals do live in smaller groups in like communities rather than just being spread out completely. So it's likely that at least some of those animals are the same ones coming back, like we see here in Burroughs Pass, especially if you're seeing them all the time. Now, are all of them the same? I don't know, but I would think that you're probably seeing some of the same individuals.
And follow up, do they know, are they family units? Or
we have no idea about their social structure. So my work in the Bahamas was all about social structure, so that's what I'm really interested in. They do live in smaller groups, but we have no idea what those groups are. Lots of times you see a male and a female and a calf, and then looks like an escort. But we don't know what those family groups look like at all for them, if they're at all. Yep. So have any of the Burroughs animals been identified elsewhere? We do have one picture, I believe, from one of the whale watching boats that was Orcas. It was Orcas Island that I believe is the same one that we have in Burroughs Pass. That's the only one so far, because we don't have a lot of pictures from outside of our area.
Have you tried a mark recapture thing to see what your local population is?
Yeah, so the mark recapture, that one's a little bit harder because of the rules and limitations you have to have for that. I don't think we have the data set yet to be able to do that because we're just in that one spot. But I'm looking into that with a colleague to see if there's a way that we can try to figure that out now that we have more data and more pictures. Yeah.
Dulles porpoises and crossbreeding.
Dulles porpoises and crossbreeding. Yes, so, and I always forget, is it the cat, is it the
Male harbor porpoise and female.
male harbor porpoise, which kind of makes sense, right? Yeah. The male harbor porpoise, it's always male harbor porpoise with female dulls when you see the hybrids. I don't know what much has been done about that, and I don't know how many there are happening now that there's quite a fewer dulls. Maybe that only happens when there's really a lot of that interaction between the two, and there's less now. But they do have a few documented hybrids between those two species, but we don't know too much more about them. Yep.
I was wondering if the Golden Gate group that's looking down has a different success rate with their photo ID.
So, yeah, I don't know. It's hard to do success rate to some degree because some of the pictures are just not usable. So I can't say like, oh, I couldn't ID it. It's like, well, there's no way to ID it. They have over 600 individuals in theirs, but they're looking down, and so they're using mainly scars. So it's the scarred animals that they're going to be able to ID, sometimes pigmentation. So it's a little bit different, and we're working closely with them with our stuff, so we are perhaps down the line be able to compare those two.
I'm assuming success rate is how many photo ops before you get a good photo.
Right, but it's so hard to know if you're getting that individual, right? So it's like there's 5 in the past, so I got 2 of them, but maybe I only got a good series of 2, and that one picture was good out of the 5. So it's just difficult to be able to do that, but we're looking into trying to get those numbers. Yeah.
This summer, my daughter and I witnessed what we thought might have been, maybe not cooperative hunting, but like animal hunting in an area. Harvesting all outside between
Oh, between harbor seals and harbor porpoises, cooperative foraging. Not seen that. We see seals all the time. They don't really seem to interact too much in our area, but not to say that they couldn't be doing that somewhere. I mean, they're both intelligent animals, so that would be really interesting to look at. Yep. Oh, those could most likely probably are some of the same individuals that we see. Yes, please do. Absolutely, that'd be amazing. Yeah. Rosario Head is also really good. But Bowman Bay, Rosario Head is a good place to see them as well. So that's what we want to find out. Are those animals, are they the same ones? Yeah, absolutely. Yep. Oh, last one.
One more. You give them names. You also give them numerical designation.
What is it?
You give also numerical designations type of name.
Oh, numerical versus names. So that's a hot topic with researchers, whether you give them actual names versus numbers. I've always been on the naming part because it's easier to remember than like T145 or 3600, whatever. But because of the amount of animals that we may end up getting, we may have to start just putting numbers as a base for all of them and naming the ones that we see more often. But right now we're just naming them. Yeah. Thank you very much.